I'd like to welcome you all to the Chemistry Senior Capstone Talks. We have 23 talks that are going on this term, and Dan represents just number 8. So that means that you have just this huge opportunity to see a whole bunch of different talks. And I want to point out to you that you can see these talks. Number one, if you can't make it, it's live streamed, and it's also available on our very own YouTube channel. So if you go to Chemistry Senior in Lawrence, you can pull up all the YouTube videos. If you'd like to come see them live, they're available. You can see them up on the calendar and when they're going to happen. In the next 2 weeks, we will have 12 talks. So basically, one talk every day of the week except for Mondays. So the next talk will be Matt Brown Tuesday afternoon. So I invite you guys to come on down and join more of these talks. Today we have Dan. Dan's been an advisee of mine for a number of years. He saw the light after being in the conservatory for a number of years. He is what I call a reformed Connie. So he decided to go the more difficult route by becoming a biochemistry major. The conservatory was way too easy for him, not taking enough of his time. And doing that, he then subsequently also fell in love with neuroscience and psychology, and that I've been working closely with him on his reform. You notice he doesn't have that strange Connie tick anymore, which is really good. He worked really hard on it. And so he's in great shape from that standpoint. He's going to be graduating this year, of course, and that he's going to do a gap year, and then after that is going to be moving on, looking towards graduate school in neuroscience. Without further ado, I'll turn it over to Dan.
Hi, I'm Dan Larson, and I'll be presenting on alcoholism and its relation to the serotonin receptor system. To start, I'd like to define addiction. An addiction is a repeated behavior with a few different stipulations. The first is engagement to achieve appetitive effects. The second is preoccupation with behavior, probably most hours of the day. The third is temporary satiation with the behavior, which returns to appetitive effects. It's a downward cycle from there. The fourth is loss of control. And the fifth is suffering the negative problems that they face. And addictions are sustained biochemically through changes in gene expression and neurotransmitter imbalances, and psychologically through positive and negative reinforcement. The DSM is the Diagnostic and Statistical Manual of Mental Disorders. They have 11 criteria for alcohol use disorders, which we colloquially call alcoholism. I have 8 up here, mainly for space reasons. Some of them are strong craving, lots of time spent obtaining or recovering, unsuccessful efforts to cut down, affects daily activities, and use continues despite known problems. There's 2 types of alcoholics. Type 1, it begins after age 25 and is associated with mild psychological dependence and drinking 3 or so drinks per night because they need to unwind in that way. And they typically don't have any other drug addictions and aren't in trouble with the law. Type 2, they sometimes are in trouble with the law. They drink a lot more. They don't lose control when they drink, though, and they're many times addicted to other drugs. And then, depending on how many criteria one satisfies, can either be mild, moderate, or severely affected by alcohol use disorders. And how does alcohol affect the brain? It activates the reward pathway. In the short term, dopamine, serotonin, and norepinephrine are released. This is responsible for the buzz you get during that first drink. And long-term effects in the same night, those neurotransmitters are not released. In fact, there's less activity in the brain, the prefrontal cortex and cerebellum, and mind in general. You get fatigued and want to eat some pizza and pass out. But chronic long-term effects include altered gene expression, changes in serotonin, GABA-A, and NMDA receptors, as well as changes in the endocannabinoid system and reward pathway, which are already implicated in many addictions. what is the reward pathway and its components? The central core reward pathway is the ventral tegmental area to nucleus accumbens to prefrontal cortex. But recently, researchers have been talking about the extended amygdala reward pathway, which includes more regions than these three, but also it includes the amygdala and hippocampus, hypothalamus, and dorsal raphe nucleus. The amygdala and hippocampus are responsible for the keeping of drug-associated memories or alcohol-associated memories. So let's say an alcoholic was driving down the road, not drunk, but driving down the road. They see a billboard for Jack Daniels and think, man, I could go for some Jack right now. That would happen in the amygdala and hippocampus. And for the hypothalamus, it regulates different changes in body state. So if someone decided to get rid of their hangover by having a Bloody Mary, that would most likely start in the hypothalamus. And then the dorsal raphe nucleus is the storage center of all serotonin before it goes to any region in the brain and body. And I have a diagram of brain here with those areas highlighted. That's the prefrontal cortex. And then here we have the ventral tegmental area, nucleus accumbens, and prefrontal cortex. That's the core reward pathway. The hypothalamus is next to the ventral tegmental area. You've got the hippocampus and amygdala here, the dorsal raphe nucleus, and then the locus coeruleus, which is the storage center for all norepinephrine or adrenaline. Here we have some dopamine and serotonin pathways. The dopamine pathways are responsible for reward, motor function, concentration. Serotonin pathways are responsible for mood regulation, memory processing, sleep, and cognition. And there are actually lots of serotonin receptors in the dopamine pathway that regulate it daily. And as you can see, they go to some of the same areas like the nucleus accumbens, the prefrontal cortex, as well as the striatum. The main point here is that they interact, and this is shown through a diagram of interactions in mouse brains that were drinking alcohol. We see serotonin goes from the dorsal raphe nucleus to the prefrontal cortex, nucleus accumbens, and ventral tegmental area, the core reward pathway, as well as the amygdala. And there are other neurotransmitters, GABA and glutamate, that also are responsible for this. So it's not just serotonin, but I'll be talking about serotonin and serotonin receptors. So before I talk about serotonin receptors, I'll go over serotonin neurotransmission. And what that means is serotonin release and serotonin being bound by a receptor. That happens when an action potential, which is an electric charge, travels down the axon in the brain, which opens calcium voltage-gated ion channels, which cause these vesicles containing serotonin molecules to move down to the synapse where they release the serotonin. The serotonin is bound by the receptor, and then that can cause many different cascades of events. Over time, chronic alcohol abuse changes these events in order to reinforce the habit. And then an agonist is a chemical that activates a receptor to produce a biological response. Serotonin is an agonist of a serotonin receptor. It's probably the simplest example. An antagonist is a chemical that blocks and competitively binds to the receptor and blocks the effect that the agonist produces. An example of an antagonist is the drug naloxone, which is used for heroin overdoses, and it competitively binds to the opioid receptor, pushes heroin out, but has no pain-killing effects, and it saves many lives. A ligand can be an antagonist or agonist. It just means something that binds to a receptor. I'll be talking about 5-HT1, 2, 3, and 7 receptors. There's many different ones, but I'll be talking about just those 3, or those 4, and a little bit about 5-HT7. It's interesting, in general, agonists for the 5-HT1, 2, and 3 receptors produce anxiety-like symptoms, and antagonists produce more calming symptoms. And excessive binding of agonists to the 5-HT3 receptor can cause one to vomit. So over time, if you drink too much in a night, that vomiting might be in part caused by binding of the 5-HT3 receptor. In addition to those 4 receptors, I'll also be talking about the serotonin transport protein, which moves serotonin from the dorsal raphe nucleus to anywhere it's needed, and also histone deacetylases, which are involved in gene silencing, and I'll go over that more in a little bit. First study I looked at, they did human post-mortem autoradiography of alcoholics, and quickly, autoradiography is when they use a radioactive biological molecule, put that in the system and let it circulate, and it emits neutrons and radiation that we can take a picture of and see how the serotonin transport protein is binding to it. They use hydrogen-3 citalopram, which is a selective serotonin reuptake inhibitor or antidepressant, so we know that the transport protein will bind to it. What they found was there was some decrease in the caudate and putamen, which assists with executive functioning, in type 1 alcoholics. They found that chronic alcohol use decreases binding in the nucleus accumbens, which is to be expected as chronic alcohol abuse is associated with lower serotonin levels and depression. They didn't find a significant decrease in density in the superior frontal gyrus, which is involved in self-awareness. Basically, what this means is that with the regions they hypothesized to be affected, not all of them were affected in the same way, or affected at all, in the case of the superior frontal gyrus. With the 5-HT1B receptor, it's present in hippocampus, prefrontal cortex, ventral tegmental area, and nucleus accumbens. Those regions are all part of the extended amygdala reward circuit, and it interacts with not only serotonin but other neurotransmitters. And as I said, when it is bound by agonists, it can create depression and anxiety-like symptoms. Polymorphisms in the 5-HT1B receptor gene are associated with alcoholism. The polymorphism is when 2 single DNA base pairs are switched, and sometimes, since we have so many DNA base pairs in the body, sometimes it doesn't do anything, but other times it can create problems. Receptor knockout mice, which are mice that are bred to not have the gene for the receptor, showed increased consumption of alcohol, and partial and selective antagonists and some agonists showed reduced consumption. So the 5-HT1B receptor has a role in modulating drinking behavior, but it's not clear which kinds of molecules will reduce or increase. The 5-HT2C receptor is present in high concentrations in the nucleus accumbens and regulates the dopamine reward pathway. They found in the brains of mice drinking alcohol expression of 5-HT2C mRNA in the nucleus accumbens as well as those other 3 regions. And they found that antagonists decrease drinking behavior. The majority of the drugs they tested were antagonists with the 5-HT1B receptor, so that's also true for the 5-HT2C receptor. In addition to that, they also found mRNA for the 5-HT7 receptor expressed in the dorsal raphe nuclei and nucleus accumbens, but 5-HT7 antagonists did not reduce drinking behavior. The 5-HT7 receptor is associated with learning, mood regulation, and sleep, so possibly in this case it might have just been responsible for instantaneous serotonin release. The 5-HT3 receptor is expressed in cortical regions, meaning regions in the front of the brain, the frontal cortex, prefrontal cortex, and also in the ventral tegmental area and nucleus accumbens. Chronic use of alcohol permanently lowers 5-HT3 neurotransmission, and lowered 5-HT neurotransmission is associated with depression, which we know can be a result of chronic alcohol use. In addition, there's an inverse relationship of 5-HT release from the 5-HT3 receptor to concentration of alcohol. So, as I was saying earlier, serotonin's released a lot in that first drink, but is not released a lot on the 8th or 10th. 5-HT3 receptor antagonists reduce alcohol intake in mouse models. So we see that antagonists for the 5-HT1B, 2C, and 3 receptor in general can reduce alcohol intake in mice. How is long-term physiology and gene expression controlled? It's controlled by histone acetyltransferases and histone deacetylases. But before I go over that, I'm going to go over the central dogma of biology, which is DNA gets transcribed into RNA, which gets translated into protein, and gets transcribed by RNA polymerase, and then ribosomes read the RNA and create specific amino acid sequences that fold to create proteins. Every serotonin receptor is a protein. There's a gene that codes for it and has it built. And histones are proteins that DNA wraps around. And I have a diagram and I'll go over this, but basically histones have a positively charged lysine at the end, and DNA has phosphate groups. The positive charge on the lysine attracts the DNA. So here, the positive charge at the end of the histone attracts the DNA, it wraps around, and then they make nucleosome, and then 30 nanometer fiber, 300, 700, and then it folds together to make a chromosome. And lysine, the positive charge on lysine is neutralized by histone acetyltransferases, which take an acetyl from an enzyme such as acetyl-CoA or another enzyme with an acetyl group, and they move that acetyl group onto the lysine, and then there's a neutral interaction between the histones and the DNA, and the chromatin, which is this, is looser, and enzymes can reach it easier, and there's more gene expression. Histone deacetylases remove that acetyl group and then leave the lysine positively charged, and the chromatin tightens up again, and there's less gene expression, what's sometimes called gene silence. Which leads me to the last paper I'll discuss, which is alcohol-induced serotonergic modulation, the role of histone deacetylases. Previous studies implicate multiple neurotransmitters such as serotonin, dopamine, GABA, and glutamate in the reinforcing effects of alcoholism. Recent studies also implicate PATs and HDACs. HDACs, antagonists or inhibitors, might play a role and be transcriptional modulators of the serotonin that's released by drinking alcohol. And they can possibly keep addiction-repelling genes turned on that get turned off when drinking alcohol increases histone deacetylase activity. Their study design involved human SKNMC neurons. SKNMC neurons are easy to grow, that's why they use them. And they treated them with ethanol in increasing concentrations of weight by volume. And then trichostatin A was their histone deacetylase inhibitor. For that, they pre-treated it with 50 nanomolar TSA and then 20 nanomolar ethanol for 24 to 72 hours. Then the RNA was harvested between 0 to 24 hours, 0 to 72 hours, reverse transcribed to complementary DNA, and then amplified through quantitative real-time polymerase chain reactions, qRT-PCR. For histone deacetylase activity, neurons were harvested in between 24 and 72 hours, and they used a fluorometric HDAC assay kit to assess activity based on ethanol or trichostatin A and ethanol. For 5-HT3 protein levels, neurons were harvested and lysed or cut at 72 hours and subjected to Western blot analysis. And then for serotonin release, the neurons and surrounding liquid were harvested at 72 hours and subjected to ELISA analysis, and I'll go over those right now. Western blot is a protein detection method involving gel electrophoresis and then transferring to another membrane, staining it with antibodies, and then taking a picture, and you can see different bands representing different base pairs. qRT-PCR is a technique for gene amplification, and the Q means quantitative, and you're seeing how much of the gene was expressed compared to how much you expected it to express. ELISA is enzyme-linked immunosorbent assay, and it involves having an antigen and then an antibody that binds to it, and where it says target protein, that would be where serotonin is in solution. And then an enzyme binds to that complex, and a substrate binds to that complex. And depending on how much serotonin is there, it'll change color, and we can graph that and quantify it and see how much serotonin concentration is present. So for the Western blot, you see the bands up here compared to the control primer. We see that ethanol increased 5-HT3 gene expression shown by protein levels, and TSA and ethanol increased gene expression even more, and we expect that because TSA leads to HDAC inhibition, which leads to more gene expression. Then we see that TSA significantly inhibited ethanol-induced serotonin secretion, and that's important because if serotonin secretion is why we feel good drinking that alcohol, it's probably the first step in creating cascades of events that lead us on the track to having reinforcement of the cravings of alcohol. If we can prevent that, then we're kind of nipping it in the bud. And then for histone deacetylase activity, we see that TSA inhibited it, as we expect, since TSA is a histone deacetylase inhibitor. With increasing concentration of alcohol, there is more histone deacetylase activity, and that has been shown in previous research and is implicated with genes being turned off, possibly genes that protect us from the addictive effects. But we see that activity is less when TSA is added. So to summarize all that, after consumption of ethanol, there's an increase in 5-HT3 gene expression, increase in HDAC activity, but TSA inhibits that HDAC activity as well as ethanol-induced serotonin production. TSA may block the turning off of certain genes that protect us by inhibiting serotonin release. Future studies should investigate the role of HDACs and HATs more with their interactions with alcohol use disorder related genes, as well as TSA and the effects on blocking 5H or serotonin receptor gene expression or repression, and they induce acetylation of transcription factors, which leads to more gene expression, most likely gene expression of beneficial genes. And then also more research should be done on medicines like naltrexone, which is already something that's currently used to reduce cravings for alcoholics. The goal should be to find medicines that target both the mechanism and the craving, and through that, combined with therapy, hopefully alcoholics can set themselves on a road to recovery. I'd like to thank Professor David Hall, Professor Stefan Dever, Professor Allison Fleshman, Professor Deanna Donahue, people in my capstone class, my friends, my family, and anyone else watching, because I definitely couldn't have done it without you guys. Here are my references. Thank you.
Questions? Wow, hands just fly up.
So have you seen any studies on tracking similar systems and what the effect of only like one drink a night or two?
I didn't encounter any, because I was mainly looking for alcoholics and the models of that, but that would be interesting to investigate and see if it takes longer to develop the addiction. Joe?
So the TSA has some kind of preventing the shutdown of genes that help regulate the serotonin system. It seems kind of like a sledgehammer approach to the problem. Have they done any research on the effects that TSA has on other gene expression? Or is this just like really preliminary?
Well, I know that TSA is an HDAC inhibitor, but I'm not sure if they've investigated it with respect to other genes not related to alcoholism.
Well, wine's got HDAC inhibitors, right? So you just have a beer and a wine and it'd be great.
Mike?
This has less to do with gene expression, but I was curious, throughout your research, if you found the reason as to why type 1 alcoholism can only be classified or can only be diagnosed in people above 25.
No, I'm not sure. Maybe they chose 25 because that's when your brain fully develops, but I don't know.
And what the difference, so there are all kinds of addictions, and that right now you're talking some sort of substance addiction, in this case alcohol. Do we see the same sort of responses and with serotonin, for example, with a gambling addiction, right? Because you don't eat money.
Yeah, well, we do see the reward pathway activated for gambling. They get similar feelings of pleasure from gambling that alcoholics get from drinking alcohol.
But do substances like alcohol or marijuana or whatever you get addicted to, does that also biochemically reinforce the serotonin release for the reward? So can you be more addicted to alcohol than gambling?
Yeah, I'm not sure if it depends. Possibly, since alcohol is something you're drinking and putting into your body, but gambling is very addictive, so I would need to investigate that.
Other questions? We'll thank the speaker again. We'll see everybody next week for more talks.